Hello, welcome to a new tutorial presented by S. Charles on cell physiology with co-reference Scholar Atlas of Physiology by Agamemnon Dipsopoulos and Stefan Sibernak. This is the cell physiology tutorial, and we are going to have cell organelles. So we start with the different cell organelles that we have in the cells. So the first point, we have the tight junctions. So these are the tight junctions here. Then we are going to have the cell membrane at this point. At this point here, we have the cytosol. At this other point here, the cytoskeleton. So we need to point out that the cytoskeleton is a part of the cell that is going to be used in anchoring the different parts of the cells. And now we have different junctions that are going to be involved, and they are going to be constituted by this cytoskeleton. So the first point is that we have this tight junction. The tight junctions are also called the zonal occludents, and these are one of the junctions. Here is the Z disc. The Z disc is also going to be made up of the cytoskeleton, and the Z disc is made up of one of the main cytoskeleton, because you need to know that there are three main cytoskeleton. We have the first one. You have three main cytoskeleton, so these are the points that we have to put in mind. And now we have this cytosol, the next part, and now you have the lysosomes. Then you have the smooth endoplasmic reticulum at this point here. Then you have the Golgi vesicles, then the rough endoplasmic reticulum. At this position here, we have the mitochondria. At this point here is the Golgi complex, which is going to be made up of different parts, as you're going to see in the next views. And here we have the nucleus, you have the chromatin, and you have the nucleolus. Now this is a view of, this view was a schematic view which that can be drawn by hand, and now this is a view that we have when we take a transmitted emission scanning. So transmission emission effect microscopy, we are going to have that this particular image. So this is according to a transmission electron micrograph. So you are going to have this image, and here is going to be the cell membrane. At this position, you are going to have the brush border for absorption. Here we are going to have vacuoles. Here we also have the tight junction. The tight junction are going to be made up of myosin filaments. So you have myosin filaments are going to be mostly used in anchoring these tight junctions, because you need to know that there are three main proteins which are making up the cytoskeleton. The first one is the actin filament, as you have seen in this diagram here. You have actin filament. The second one is the myosin filament, and the third one is the microtubule. So you have microfilament, the microtubules, and then the last one, the intermediate filament. So the microtubules are found inside the tight junction of the zonal occludents. Then you have the microfilaments are going to be found inside the zonal adherents, and lastly you have intermediate, and all the rest are going to be found inside the Z disc. So now we continue at this portion here. We have the cell border. Here you are going to have the mitochondria. At this position here, you are going to have the lysosomes. Here you have the rough endoplasmic reticulum. You have the autophagosomes, when the lysosomes have bound with vacuoles that contain food particles. And here we have Golgi complexes. Golgi complexes are going to be made up of Golgi vesicles and all that. And here we have the basal labyrinths, and here we have the basal membrane of the cell. So now we continue by transcription and translation. So in translation, the first thing to know is that anytime you want to have any genetic any functioning in the cell, any function in the cell are going to be due to the genetic control. So the first thing to point is that you need to do a transcription of that gene from the DNA, and the DNA is found inside the nucleus. So now the transcription, you need to know that the direction of the transcription occurs in the 3' to the 5' direction. So before we start knowing what's the meaning of 3' and 5', as you are seeing in this later part here, we need to first point out the constitution of the DNA. So what is the DNA made up of? the DNA is made up of one ring of ribosome. It's made up of a phosphate ion, and it's also made up of a nitrogenous base. So the first point is that the center of that complex of the nucleotide, because each DNA are made up of these nucleotides, and these nucleotides are made up of three of what I've said. I've said one ribosome ring, the second one we have the phosphate ion, and last one we have a nitrogenous base. The nitrogenous base is going to be connected to the carbon number 2 of the ribosome, and then the phosphate ring is going to be connected to the carbon number 5 of the ribosome. So now, when I mean by 3 to 5 prime direction, I'm just speaking about the ribosome carbon atoms. So the first direction is going to start from the carbon atom of ribosome 3 to the carbon atom 5. So that's the meaning of 3 to 5 prime direction. So we have different nucleotides that you can find there. The main nucleotides that you have inside the DNA are, you have number 1 adenine, the second one thymine, the third one cytosine, and the last one is guanine. So these are the four nucleotides. Adenine is going to pair up with thymine, cytosine is going to pair up with guanine, and this is how it occurs. So the transcription is start at this point here. You first start with the DNA helicase. The DNA helicase is going to splice up the particular portion of the DNA that wants to be expressed, and then later on you're going to have RNA polymerase. RNA polymerase is going to act in the 3' to 5' direction, producing a primary RNA which is going to be 5' to 3' direction, and then later on you're going to have a splicing, and that splicing is going to produce this mRNA. So now let's visualize how splicing occurs, the transcription occurs at this point here. So the first thing is that you need to know that the DNA molecule is going to be made up of introns and exons. The exons, which are in blue, are what are supposed to be transcripted, and the introns are the heterochromatin, what is not supposed to be transcripted. So now at this point here you have a transcription. The transcription is going to be producing this primary RNA which has both the introns and the exons, and then you are going to have a splicing which is going to cause the removal of these introns, and then addition of a 5' cap and a 3' poly A tail, and all that is going to produce your messenger RNA. Now the messenger RNA is going to move out of the cell. When the messenger RNA moves out of the cell, it can have different things. These different things can occur to that. The first point is that it can bind. The most important is that it's going to bind to the ribosome. And now inside the ribosome you have this part. You are going to see that there's going to be translation. The next process that is going to occur to the messenger RNA is that it's going to be translation of that first genetic component that comes from the DNA. And now you need to point out that translation occurs instead in the 5' to 3' direction. Initially I've told you that the transcription was occurring in 3' to 5' direction, and it was producing a corresponding mRNA which was formed in the 5' to 3' direction. So it implies that the translation is going to occur in the 5' to 3' direction. And now how does translation occur? It occurs inside the ribosome, and inside the ribosome you're going to have your tRNA. The tRNA has two main parts. It has the anticodon. This portion of the mRNA is called the codon part, and now the tRNA is going to have the first portion that's going to contain the anticodon part, and which is going to pair up with the codon on the mRNA, and then it's going to contain a second portion which has an amino acid bounded to it. And now when messenger RNAs, when the tRNAs are going to be connected to each other, they are going to form a peptide change by peptide bonding. So this is how transcription and translation occur. Thank you. So now the next point we are going to view is how we have protein. We have already seen the protein synthesis. we have sorting and recycling. So now, how does it sort? So when the proteins come out from the rough endoplasmic reticulum, they are going to enter in the cis terminal of the Golgi apparatus. After entering the cis terminal of the Golgi apparatus, they will sort out from the trans terminal of the Golgi apparatus. From that trans terminal, they can be packaged into secretory vesicle from the trans terminal of Golgi apparatus, and then signals are going to cause it to move out of the cell. Also, they can be functioning as receptors, as you can see from this portion here, where receptors can be binded to the cell membrane, as you can visualize here. So now the next position is that they can form these M6P receptors for recycling. So it can come out, it can be recycled, and it's going to bind with these substances that are going to come enter into the cell via endocytosis, and all of that is going to move out. So this is what is involved here. So now let's visualize the cell membrane. So the cell membrane is made up of a bilayer sheet of mostly phospholipids, and with those phospholipids we also have addition of lipoproteins. We have glycoproteins. So this is glycoproteins here, and they can serve as receptors and lipoproteins. So at this position here, we count this as this glycocalyx. This is a lipid bilayer, and this lipid bilayer can also be made up of cholesterol. So inside you have peripheral membrane. You can have peripheral membrane inside and outside. They are going to be called internal membranes here, and here are going to be called external membranes. So these membranes that are going to pass through are going to be called channel membranes. So now, how are these phospholipids formed? The phospholipids normally are formed from a triglyceride chain. Normally, you all know that the triglyceride chain is going to be made up of one main backbone of glycerol, and then 3 fatty acids are going to be attached to each terminal of the OH group of the glycerol. But now, at one position of the triglyceride, you are instead going to have a phospholipid, which you are instead going to have a phosphate ion that is going to bind at that position at one OH of the glycerol, and that is going to form what is called phospholipid. So a phospholipid is a molecule made up of one molecule of phosphate, one molecule of glycerol, and 2 molecules of fatty acid. So inside the membrane, you can still see cholesterol here. You have the phospholipids, you have the glycolipids, and you also have the proteins inside. So these are different things that you can see. Now we are going to see now calcium ion transport inside the endoplasmic reticulum. So now you need to know that the calcium ion has the calcium ion ATPase, and the calcium ion ATPase causes the calcium ion to enter inside the endoplasmic reticulum, and this is going to be very effective when you are going to see the contraction of muscles in the next chapter. So now we are going to see calcium ion enters here via the calcium ion ATPase, so inside the endoplasmic reticulum. And now when calcium ion comes out, you see there's only a single of depolarization that is going to cause calcium ion to come out, and it comes out via the calcium ion channels. You see that the names of the calcium ion channels are going to differ from if it is skeletal muscle or smooth muscle. In skeletal muscle, it's going to be called Ryandine receptor channels. So now we continue with the mitochondria. So now in the mitochondria, now we have the mitochondria is going to be made up of outer membrane, inner membrane. We have the matrix. Inside the matrix, there it occurs, the Krebs cycles occurs there. Inside the membrane, we have the electron transport chain, as we see in this diagram. But now the explanation of electron transport chain is going to be given in the next chapter, respiration. Okay. So now let's continue now with the gap junctions. So we need to know that we have these different proteins inside the gap junction. The gap junction is going to be mainly made up of connexin proteins. We have connexin proteins, and we have this protein, and we know that every time we have a protein, we have an NH terminal and we have a COH terminal. So these are just gap junction. They are mainly made up of connexin proteins. And now let's see other junction. I've initially told you that when you have the cytoskeleton is going to be made up of three main fibers. You have microtubules, you have microfilaments, and you have intermediate filaments. The microtubules are what are mostly going to make up the junction, the tight junction. The microfilaments are mostly going to make up the zonal adherents. And lastly, you have intermediate filaments. Intermediate filaments, they are in addition to the microtubules and the microfilaments, are going to make up the Z-gis, which is one of the most strongest junctions found inside the cell, and those cells are mostly the epithelial cells. And this is why these cells that are taken as our reference point of study of the cytology being epithelial cells, you see that there are many junctions found here. So now we continue by the fact of diffusion. So we define diffusion first as the movement of ion particle from a region of higher concentration to a region of low concentration down the concentration gradient. And now this is due to the Brownian particle motion. You see that all the particles are going to move randomly, and now in cases of high concentration it's going to move now in cases of low concentration, as you can see here. So now this is how diffusion occurs. And now diffusion respect the formula called fixed formula, as you can see in this position here. So now fixed formula is defined as the rate of change of diffusion, that is the number of moles that is moving per unit time, is equals to the area times the permeability constant. This G is taken as the permeability constant times another constant, which is the diffusion constant. We have the permeability constant, which depends on the membrane. We have the diffusion constant, which depends on the molecule. We have the surface area times the surface area and times the difference in concentration of the two medium divided by the thickness of the separation of the medium. So this is the formula called Fick's law. So Fick's law can be shown here again. Here P is given as a permeability constant, and that permeability constant is depending on the membrane. We also have the diffusion constant. So all this formula is given here. So we have K is the permeability constant, A is the surface area, D is the diffusion constant. This is the change of the difference in concentration of the two medium, and this is the difference in thickness, the thickness of the medium. So we thank. So this is the next part. So now the next part now that we are going to visualize is osmosis. So now osmosis, as you can visualize here, you still have the rate of change of osmosis from point to point. And now for osmosis, which is a number of moles of water molecule, flowing from higher potential to lower potential divided by time, you instead have a certain constant, the filtration constant, times the osmotic pressure difference between the 2 points. So these are the different positions, different things that you can visualize here. And now the next one, we have the sodium ion potassium ion ATPase. So the sodium ion potassium ion ATPase, for the sodium ion, you see that one sodium ion, three sodium ion moves out, and then two potassium ion move inside. So that's the sodium ion potassium ion chain. So now the next point we need to visualize are the transport method inside the cells. You need to know that the transport method inside the cells are, we have visualized diffusion, we have visualized osmosis, and now the next one that you can see now is going to be active transport. Active transport now is the inside the movement of particles from a lower concentration to a higher concentration, up the concentration gradient, and while using energy. This energy can be directly using ATP or indirectly using ATP. When it is directly using ATP, it's called primary active transport, and when it is indirectly using energy, it's called secondary active transport. Example of primary active transport are going to be calcium ion ATPase, sodium ion potassium ATPase. So where the ATP is going to bind directly to the carrier molecule and is going to cause the movement of the ions. Now we have secondary active transport. In secondary active transport, we have the movement of one particle which is moving down its concentration gradient, while the other particle is moving up the concentration gradient. The energy comes from the particle that's moved down the concentration gradient. So an example is going to be symphotas and antipotas. Symphotas, one molecule is both of the molecules are going to enter or move out of the cell, while antipota, one molecule enters while the other molecule move out. So you can see from here we have sodium ion, glucose, symphotas. So all of them are going to enter. So sodium is moving up the concentration gradient, the glucose is moving down the concentration gradient, and then you are going to see how it's going to enter. Then you have the next one, the antipotas. So we can visualize them. So the calcium ion, hydrogen ion, antipotas. So from here we can also see the cell migration here. So we have here is going to be exocytosis. So now for exocytosis to occur, we need to know that it first starts from the Golgi apparatus when you have the packaging. So when you have the packaging, the coating at the trans-terminal of Golgi apparatus is going to occur via the cotoma coated vesicles here, as you can see here. These cotoma coated vesicles are going to bind now with the cell membrane, and they are going to be with the T snare on the as a receptor on the cell membrane, and this is going to be lost via exocytosis. Then the next point is going to be cell migration. Cell migration just occur when we have the polymerization of the actin filament. This polymerization of the actin filament now is going to propagate the fibers, the cytoskeleton, toward one part of the cell, and that propagation is going to cause the movement of the content of the cell toward the other part, as you can see from this diagram. So now the next point that you are going to see in the cell physiology is going to be the electrical membrane potential. So the electrical membrane potential can be given from the Nernst law. So the Nernst equation is going to be given as the electric potential is equals to the RT all over Fz ln of the concentration of the ion outside the cell divided by concentration of the ion inside the cell. So now example, so this Nernst equation can give you the electric potential that you can have in each cell with respect to their concentration. So now we usually know that the resting membrane potential of a cell is given as negative 90 millivolts. But now why is it that the resting membrane potential of the cell is going to give it as negative 90 millivolts? This is because the cell is only permeable to potassium ion, and now when you calculate the Nernst, using the Nernst equation, the concentration of potassium ion outside the cell and the concentration of potassium ion inside the cell, you are going to realize that the value that is going to come out, E, is going to be exactly negative 90 millivolts. So there is a certain position here that I want to show you. The concentration of potassium ion, if you use all the points that have the R, which is going to be equals to 8.314, you use the T, the temperature in a body, which is 37 degrees Celsius, you convert into kelvins, and then divide by the F is going to be just the number of coulombs. Now the usual is given as 96,500, and then you have N of the potassium is going to, potassium is charge of 1. So when you substitute all this, you see the concentration of potassium outside the cell, inside the cell, is going to be 140 milliequivalent per liters, and then you have the concentration of potassium inside the cell, which is 4.5. When you substitute all this, you have exactly negative 91 millivolts. So you realize that actually the Nernst equation can be used to find the electric potential of the cell when the cell is permeable to only one ion. So now this is why you can also have the Nernst equation. You see that at depolarization, you see that the neuron is going to depolarize to produce an electric potential of plus 30 millivolt, and that plus 30 millivolt is formed because of the concentration of the sodium ion, because at that level we have sodium ion which is being permeable. That is why the maximal electric potential that you're going to have is going to respect inside sodium ion. So we have visualized all the main things involved with cell physiology. So say thanks for the kind attention. Please don't forget to like and subscribe for the channel. Thank